my next speaker, I am continually A, introducing and B, apologizing to for mangling the second name. So
don't worry about it.
Yeah. Who will be speaking about, sorry, I need new glasses, modeling the application of new genetic-based tools for pest management. Aidin is from Land Care Research in Dunedin. There's a bit of Land Care Research.
Yes, thank you. Actually, Neil and Peter went through half of my slides, so not much left for me. But, okay, so these stuff, what is gene drive? So you heard a lot about it. One thing I want to point out is this phenomenon or concept of selfish genes. So these are the genes that off and on occur, or like the Mendelian genetic probability have got 50% chance of inheritance. So like the diagrams that I've got one of them that I'll show you. They dilute through the population, but the purpose of gene drive is to artificially basically create these selfish gene elements that can potentially go through the whole population, basically stay there forever. And this is basically the concept of gene drive, is to take advantage of this the attaching the genetic payload, which honestly I don't know what payload means, but it's there. So this, you've seen it for a million times. As you see, the purpose of gene drive is to prevail the bad gene. And we were talking with Peter, it's basically going against the natural selection. So natural selection puts the bad things, discards the bad things, but we want to make sure it prevails. So my work is on modeling these theoretically, basically. So you've probably heard of, or in biology, there is this in vivo, in vitro, in situ, this kind of models or experiments. We've got this as in silico, which happens in a CPU of a computer. So we've looked at simulating these gene drive situations in population models. So common population models you might have heard, like logistic models, or discrete or continuous Markov chain type models, where the population, an individual in a population is at a certain stage at any time, and then it moves to other states, basically, I should say, not a stage. And the rate of transition from one state to other is determined by either a rate or a probability, or matrix population, Leslie matrix. I think ecologists and demographers work a lot with this kind of models, where the population is structured. You either have a gender structure or age structure. So we thought to, mash these together, matrix-based population model with a Markovian transition rule. So something like that. So this is quite an old population model used using matrices. So as you see, we've got you can be a female, you can be an adult female or a juvenile female. Same thing with the male. You've got the zygote state Z, which divides into males and females according to a sex ratio. Then at some certain rate, the juvenile grows and becomes, matures and becomes an adult. Then adults have a survival rate, and given some birth function or marriage function, they produce these zygotes. So I promise this is the only math I'm going to show you here. So this is the birth function we used. So the minimum of M and F, that is actually quite a powerful marriage function. That here we're using a monogamous one-to-one sort of a mating strategy. But in other types of mating can be accommodated with this simple model: polygamous, polyandrous, multiple wives, multiple husbands. So this is that minimum of M and F gives you the number of matings or number of marriages. You multiply it by litter size and then normalize it by population and use it. And then with the matrix population model, the model gets updated at fixed time intervals. So it's a discrete model. In terms of discrete in time. So we basically update our model monthly. Okay, now things that we have planned to explore, this is from the Champer et al. Paper 2016. So you see there is a list of basically types of gene drive systems. I'm going to talk about X-Shredder and Medea, modeling these two. So this is the idea of X-Shredder. So as you see, the name has it, it shreds the X chromosome. And so the idea is that it cleaves the X chromosome to a point that it gets destroyed, and the sperm will only carry the Y chromosome. Therefore, when the mating happens, the male sperm only contributes Y, and all the offspring, they are male. And therefore, this causes a basically sex distortion in the population. You get to a point where you got too many males and not many females, and the population collapses. So this is what happens in the wild in a normal situation. And with X-Shredder, you see the sperm in both cases contributes Y, so the population becomes filled with male offspring, basically. And this is, again, on the table. So this is, the X-Shredder, the drive can be assembled. This is, again, my language. If it's not kosher enough, I apologize. But it's assembled either autosomal, somewhere else, or on the Y chromosome. So this is the one on the Y chromosome, which they also call it Y drive. So as you see, that red Y is the one that has the X-Shredder on it. And you see that the frequency increases. And if you look at the ratio of male to female, is already 3:1. So that's a promising start. And here is the model results. So you see the blue curve, or the red curve, is the total population. The blue curve is the wild type normal population. We've let it reach its steady state, and at around about time, 100 months, we release one of these males. So there's no need for females, basically. Obviously, females don't have the Y chromosome. So one of these males, and the males grow slowly, and you see what happens. The population basically collapses, and the plot on the right is the female to male ratio, which starts at 1:1 in the beginning and goes to 0. So in theory, it works. Now, Medea is a little bit more complicated than X-Shredder. What happens here is, now this one is basically the mother is responsible. It's kind of like that mother's curse, or TFT, Trojan female technique idea, where, as you see, zygotically expressed antidote in the survival of 50% of embryos, but 50% of embryos that originate from a mother, from a heterozygote mother who is bearing this Medea gene or whatever. There's another one similar to this called Medusa. So I don't know, somebody making up these names. So I don't know, is that clear enough? So what's happening now? If the Medea-bearing mother mates with a wild-type male, then half the offspring will die because they will be homozygote wild-type, so they won't carry the Medea element. And if a Medea-bearing mother mates with a Medea-bearing male father, then 25% of the offspring will die. So I've got some tables here that should make it more clearer. So what we have is this, So here is the case where Medea-bearing mother mates with a wild-type male. So you see these two, these are the wild types, the plus, because they didn't inherit that M from that mother, therefore they will die. These ones here is, father is Medea, mother is wild type, so nothing happens. So these wild types survived because their mother was okay, was all right. But here you see they're both Medea-bearing, and that's why 25%, a quarter of the offspring, which is the wild type plus, they will also die. Okay, now, this is not designed to collapse the population. This is more a replacement strategy, so a population replacement strategy. So that's basically in a nutshell what happens. Wild type offspring whose mother had M element won't survive. So all the wild types whose mother is M, they will die. Okay, and that's what happens. So the red curve is the homozygote M. So we get a little bit of these in the population. The green is, so as you see, the wild type population is completely removed, and instead it's replaced with this new, you can call it, I don't know, transgenic females or whatever. So this is basically a dream to sort out malaria or dengue, so that you replace the bad females, the nasty females, with transgenic one, with the good ones. Okay, so future work, a lot of work can be done around this, looking at more genetic-based techniques and modeling different hypothetical scenarios, modeling resistance, modeling if the effect is partial. So here we've assumed the effect is 100%, but sometimes it could give, you know, half of those could die, half survive, you know, this sort of stuff. But this is, again, a big project when we incorporate a spatial structure into our model. Dispersal. So imagine you've got an island and we release those extra in one corner of an island and watch them spread, basically. That's the team I'm working. So Hamish Spencer, I think, known to most of you. David Bryant, he's a mathematician too. Neil and Dan is my boss. And thanks to National Science for funding my job, basically. Thank you.
Do we have any questions for him? Not leaving until we see any questions. So how complex do you think these models need to be, and if we're talking about modelling population collapses possums in the field?
Depends on how much detail you want to. So this is the simplest we've started with, simple and robust, something that works. But the more detail you want to incorporate, and also CRISPR or things that we're going to do later, it's not in molecular level, it's all in population level. So no biochemistry, basically. But again, depends on how much detail you want to include in the model. Then the model gets more equations, more parameters.